Good morning children, I am Sarga SS. Now I am going to teach you DNA replication. As you know, DNA replication is the process of formation of a new DNA molecule from the pre-existing DNA molecule. So what is DNA replication? Formation of a new DNA molecule or DNA strand from the pre-existing DNA molecule. So, this DNA replication was first proposed by Watson and Crick in 1953. Watson and Crick in 1953 proposed a scheme that DNA replication is semi-conservative. DNA replication is semi-conservative. That means half is taken from the old one. That is called as semi-conservative. And according to the scheme proposed by Watson and Crick, the double helix or two-stranded DNA can separate during replication. So the two strands, the double helix strands can separate during replication, and if this is 5 prime, then 3 prime. Okay, 3 prime, here 5 prime, then 3 prime. So here this can separate into two strands, and each strand acts as template strand. So this one, the first one, 5 prime, 3 prime, can make a new strand. I'll show you. So here they can make a new strand, and the second one here also the second strand, 3 dash or 3 prime, 5 prime, they also can make a new strand with the polarity 5 prime, 3 prime. Is it clear? So, according to the semi-conservative manner of replication, DNA replication, the scheme proposed by Watson and Crick, the two strands of DNA can separate and each strand acts as a template strand. What is meant by template strand? I taught you in the last class. The strand which can make a new strand, which is capable to make a new strand, that strand is called a template strand. Here the two strands can act as template strand. So here this is one template strand, here we can see, and this is another template strand, here we can see. The two strands, the two separated strands of double helix DNA molecule can form a new strand which is complementary to the old strand. Is it clear? So after replication, the DNA strand consists of one old strand or one parental strand and one newly formed strand. Is it clear? So once more we can see the scheme proposed by Watson and Crick. During DNA replication, the two strands can separate and each strand acts as template strand, and they form a new strand which is complementary to the old strand. So after replication, the DNA strand consists of a parental strand or the old strand and a newly formed strand. I'll show you that. So suppose this is a DNA strand. So this red color represents the new strand. So after the two strands can form a new DNA strand or DNA molecule. So this type of replication is called a semi-conservative replication. Is it clear about semi-conservative replication? Next we can see two scientists, Meselson and Stahl, conducted an experiment to prove that semi-conservative or DNA replication is semi-conservative. So next we can see proof for semi-conservative mode of DNA replication. So here this experiment is also known as Meselson and Stahl experiment, and it was conducted by Meselson and Stahl. And the experimental material scientists to prove that DNA replication is semi-conservative. In semi-conservative manner, the DNA can replicate. Okay, and they grew E. coli bacteria in a medium containing ammonium chloride. Ammonium chloride. Okay, for several generations. So the experiment, the first step was they grew the Escherichia coli bacteria in ammonium chloride whose nitrogen is a heavy isotope, N15 is a heavy isotope of nitrogen. Okay, then observe several generations, and this E. coli can replicate in every 20 minutes. So they were observed at various time intervals. And after that, the newly formed strand, after 20 minutes, they observed the first generation. They observed that the newly formed strand consists of N15. That means the N15 got incorporated into the DNA strand. So this newly formed strand, the two strands are incorporated with the N50. That is a heavy DNA molecule because it contains heavy isotope of nitrogen. So this is a heavy DNA. And then it can be separate. So several DNA are there and some of them only incorporated with the N50. So this N15 or heavy DNA can be separated or differentiated from the normal DNA by the process of centrifugation. Centrifugation in cesium chloride. Cesium chloride density gradient. Cesium chloride density gradient. So why this is centrifuged in cesium chloride? To differentiate the heavy DNA from the normal DNA. So after centrifugation we will get they got all the newly formed DNA with the heavy isotope of nitrogen. Is it clear? They were collecting all heavy DNA by centrifugation in cesium chloride. Then what they are doing next? So here, as I told you, they grew the E. coli in N15 medium. Then now they collected this bacteria with the DNA which is having heavy isotope of nitrogen, or we can say heavy DNA. So this is a heavy DNA as it incorporates with the N50 DNA. Is it clear? Okay. Now, after that, they put this heavy DNA into a medium which is containing ammonium chloride, but the nitrogen is light. Okay, so here the nitrogen is heavy isotope of nitrogen because N15 is there. From that medium, they were collected this heavy DNA. Heavy DNA means both the strands are incorporated with the N15. And they were collected such heavy DNA from this medium. Then it allows to grow in a medium which contains N14. N14 means it's a light isotope of nitrogen. Okay. After that, they observed that. So as I told you, so here this is N15 and here this is N15. The two strands are N15. We can separate the two strands. So this is N15 and here also this one N15. And after 20 minutes, as I told you, every 20 minutes the DNA can replicate, the E. coli can replicate. So by putting into light isotope of nitrogen containing medium, after 20 minutes they observed the first generation. And they were noticed that a new DNA or newly formed DNA which consists of a hybrid variety. Hybrid variety means which contain the two strands. One is with the N15 and another one is with the N14. And another strand also, they got separated, then form a new strand, and that new strand is having light DNA or light nitrogen containing DNA strand. And this variety or this DNA is called a hybrid DNA. Why this is called a hybrid DNA? Because it contains N15 and N14. Okay, so here this N50 we consider this as heavy DNA. So this N15 and N14 together we consider this as a hybrid variety. Two different varieties are there. So clear? Then again they observed after 20 minutes. Okay, so here we can have, see first N14 and N15. After first generation they got N14, N15, a hybrid variety. Then F2 generation, that is another 40 minutes or 20 minutes, that is total 40 minutes. Here after 20 minutes. So as I told you, after 20 minutes we can have a hybrid variety. This variety is called a hybrid variety or hybrid DNA. And after 40 minutes or in the second generation, they got N14, a light DNA. Here this is light DNA. Light DNA and hybrid DNA. Hybrid DNA. How this happens, I'll teach you. Okay. So first we can see the N15. First, the N15, when it grew in the medium containing N14, what will happen? So, this strand which contains N15 get incorporated. I'll show you that. So, these also N15. And after 20 minutes, a new strand is formed from the template strand, and that is having light DNA. Here also they are having light DNA. So this is the hybrid variety. This one hybrid DNA and this also hybrid DNA. And this was happened after 20 minutes. Here we can write after 20 minutes. Okay. Then in the same medium, N14 medium, which is having the light nitrogen, they observe after 40 minutes. 40 minutes. The medium is same, which is the medium N14. Okay. So here we can separate these two first. The first one is the N50, and the second one is N40. So we are considering this one first, this strand first. So they get separated like this. Then this strand going to form a new strand, and that strand will incorporate with the what? Here N14. Okay, and this strand also form a new strand which is also incorporate with the N14. So clear? So this strand is having N15. The parallel strand is with the N15 and the newly formed strand is with the N14. Here I will write N15 first one, this one N14. So this is the hybrid DNA. Hybrid DNA. But here, this is a parental strand. Parental strand is having N14 and the newly formed strand is also having N14. So this is not a hybrid variety or hybrid DNA. This is the light DNA. So clear? And the first one is heavy DNA. Okay. So heavy DNA, when it is allowed to grow in the medium which is containing light, nitrogen, after 20 minutes they replicate, and while during replication the two strands act as template strand. Here the two template strands form a new strand and both the new strands are incorporated with the N14. So they got a hybrid variety or hybrid DNA which is having N14 and N15. Okay, so this is the heavy DNA strand and this is a light DNA strand. And after 20 minutes or total 14 minutes, they observe that in the same medium, the first strand gets separated and they undergo replication and form new strand. Again, this we can consider. How we can consider? The same will happen here. This one form a new strand which is having N14 and this one will form a new strand, this is having N14. So this is a hybrid variety and this is a light DNA. Is it clear? So in the next generation or second generation, they got equal amount of hybrid DNA and light DNA. Is it clear? Then I will show you the picture. Here the first generation, they got hybrid variety which is having N14 and N15. But after 14 minutes in the F2 generation, they got light DNA and hybrid DNA in equal amount. That means, so this one is from the parental strand and then the new strand. Here also, this is from the parental strand. One strand is from the older strand or parental strand and one is the newly formed strand. This is the semi-conservative. Semi-conservative means half or semi part of the DNA is from the old one or parental strand. That is the meaning of semi-conservative manner. Okay. Here also you can see the N14 in green color and 15 in purple color. Okay, so this is a hybrid variety. When after 40 minutes, we can see this is after 20 minutes. This is the first generation. First generation all are hybrid. Hybrid means here this is N15 and this is N14. Here also like this. But after 40 minutes in the second generation, along with the hybrid variety, light DNA also they got. This is also light DNA. Light DNA means the DNA strand incorporated with the N14. Is it clear? And this provides an experimental proof for semi-conservative manner of DNA replication. And another experiment was conducted by one scientist, Taylor and his colleagues, by using radioactive thymidine. In fava beans. And this experiment also provided evidence or proof for the semi-conservative manner of DNA replication. Hope you understood everything about DNA replication and the semi-conservative manner of DNA replication. That's all for today. Thank you.